what you need is for those molecules to somehow get together and react. That's what we want. Our, our ATP isn't just going to make itself. It requires the energy of glucose being broken down. Glucose isn't broken down just by itself. It needs lots of other chemicals in between. So the whole breakdown of glucose in cellular respiration, the only reason it works is because oxygen is so reactive. Oxygen is so electronegative. It draws electrons towards itself. It oxidizes glucose, breaks it up, steals its electrons. Um, and yeah, sure, that's spontaneous, but if it were just left to its own devices, it would happen so slowly that it wouldn't even be wouldn't help us at all. And the reason it would happen so slowly is what are the chances of all of those molecules coming together at the same place at the same time? You know, it's as probable as getting a date on a Saturday night in a club. You know, another terrible analogy. So, how can we speed things up? Well, we do it by a catalyst. There are other catalysts other than enzymes, but enzymes are catalysts. In this case, it's a chemical agent that speeds up a reaction without being consumed. So that is an enzyme, a catalyst, chemical agent. Other catalysts are like temperature. If you increase temperature, then the molecules will move around more quickly and they're more likely to collide with one another and react. But an enzyme is a chemical catalyst, a catalytic protein coded for by our DNA. Some diseases occur as a result of a lack of some enzymes or too many enzymes. So every chemical reaction between molecules involves bond breaking and bond forming. Something generally breaks, something generally forms. When you're pairing exergonic to endergonic reactions, both of those things happen. So what is an activation barrier? It's basically the barrier to those chemicals coming together in the right place at the right time. So that's an activation barrier. So what has to happen for a chemical reaction to take place? Well, one type of chemical reaction is hydrolysis. That's spontaneous digestion, just breaking things down. Here's sucrose. It gets digested by sucrase, very nicely named enzyme. Ase, a bit of water, hydrolysis, and it breaks it up with one hydroxyl molecule attaching to one and a hydrogen to the other, breaking it up into glucose and fructose. That's what your table sugar is, sucrose. It's a combination of glucose and fructose, hydrolysis. But it's just not going to happen unless they're in the same place at the same time. So activation energy is like making molecules a bit more unstable or bringing them together or providing an environment that's conducive to the reaction. Yeah, so it's the initial energy needed to start a chemical reaction. Sometimes, yeah, it's heat. Heat can start chemical reactions happening, but generally far too slowly for life. So here's an energy profile for an exergonic reaction. So here are your reactants. You know, they have bonds here that need to be broken. The transition state between the reactants and the products is such that, well, something has to happen for these bonds to break. It could be very hot, in which case they'll be moving around so much and the bonds really aren't that strong that they will just simply break. So that could happen. And then we have the products. Of course, they're more stable and it's happened spontaneously. We can tell that because the change in G is less than 0. That's called an energy profile. So this transition state, in order to get those molecules to be unstable enough to split, that's called the transition state, and the energy required is known as the energy of activation. So often you have to put in a little bit of energy just to start something off. But overall, it's totally exergonic. How do enzymes then lower that barrier? They can lower this activation barrier. So this would be without an enzyme, it's quite a high barrier, but with the enzyme, it's less. So it's more likely to occur with that enzyme, and of course at a greater rate, which is what we want. So how does it do that? Well, we'll get to enzymes in a moment. We can see how the enzyme actually lowers that activation energy. Usually it's just by bringing them together. So one thing about enzymes, they're very specific. They're not going to go messing around with lots of different reactants. The substrate, that's the reactant that the enzyme acts on. In enzyme terminology, it's called a substrate. The enzyme binds to that substrate, forming what's known as an enzyme substrate complex. And the enzyme has an active site. So this is a model of an enzyme, and here's the active site. That's the site to which the substrate, it's misspelled there, will bind. Every enzyme has an active site. So we used to say it was a lock and key model. When I went to university, it was a lock and key model. That active site was exactly right for that enzyme, but now it's not called that anymore. It's called an induced fit of a substrate. It's not a perfect fit. Sometimes it requires a bit of juggling for it to land on that site. Sometimes it lands as a result of van der Waals interactions, sometimes hydrogen bonds.
Sorry, but the same term, active site and induced fit of a substrate?
No, the active site is the physical site, and the induced fit is the action of fitting into that site.
Oh, okay. Thanks.
Yeah, good question. So bringing chemical groups of the active site into positions that enhance the ability to catalyze the chemical reaction. In other words, having certain parts of the molecules facing each other, so they're more likely to form a bond or for that bond to be broken, depending on whether it's hydrolysis or a synthesis reaction. Works the same either way. So in an enzymatic reaction, the substrate binds to the active site. So that's where the catalysis happens. Here's a terrible diagram that's not moved over very well, but
sorry,
be that as it may.
Can I ask
Sorry,
a question
Eduardo?
real quick? So what's the difference between an enzymatic reaction and induced fit of a substrate?
The enzyme catalyzes the reaction. In other words, it helps it along. And the active site is where the substrates bind.
So
and those substrates are going to go through the reaction.
Right. Is there a difference, though, between the enzymatic reaction and the induced fit of a substrate? So that's the action of fitting into the active site, right?
Yes, but that's not the reaction that's going to occur. The settling in the active site has to happen first before a reaction occurs.
Oh,
so it's
okay.
a series of events. Yeah, it
right.
is a series of events for sure. Yeah, good question.
Thank
yeah,
you.
Terrific. Yeah. So you can take a look at this in the text as well. Here's the substrate. In this case, there are two separate molecules, fructose and glucose. And here's the enzyme with its active site. Here's the active site here. And these two just happen to fit very nicely into this active site. So the enzymes do attract those molecules. They're held in site by weak interactions like hydrogen bonds and van der Waals. So because you don't want the enzyme binding to your substrate and not ever letting go, that wouldn't be any good. So the interaction between the active site and the substrate is quite weak. So how does it lower the energy activation and speed up reaction? Well, it's like a template sometimes, just kind of orienting the substrates in a conducive way. It might stress the substrates and stabilize the transition state. So if it's a case of hydrolysis, well, those or that one molecule that's going to be split might land. It's pretty stable when it lands, but the enzyme kind of goes woohoo and it causes it to be less stable. It stresses it, so it's more likely to break. Provides a favorable microenvironment. So that may be that the active site itself has some polarity about it, for example. Sometimes participates directly in the reaction. So in this case, we've got an enzyme substrate complex and the series of events. It's, in this case, stresses the bond and our products are different and they're released as separate products. In this case, it probably wasn't fructose and glucose, some other kind of molecule, because I'm seeing now that I look at the whole picture that it's an exchange reaction. Yeah, it's an exchange reaction in this case. Yeah, and you know what? This enzyme, it's unchanged. So when that reaction's all over, it's unchanged. It'll be there to catalyze more reactions. It can catalyze thousands of reactions per second. Yeah, sorry about that slide. Yeah, so it orients substrates correctly, it strains the bonds, it provides a favorable microenvironment like some low pH perhaps. It might covalently bond to the substrate briefly, thereby lowering the energy activation. Just making that reaction more likely to happen. That's all we mean by that. Yeah. And the activity of an enzyme, though, it's affected by the environment around it. Like when you consider all the different environments in the human body, even that enzymes have to work. I mean, sure, our temperature is 37 pretty much everywhere, but our pH is not the same everywhere at all. Our intestines have a basic pH. Our stomach has an extremely acid pH. And that kind of environment really affects how the enzymes can work. So optimal temperatures, yeah, for a human enzyme, it's about 37 or so. But there are bacteria that live at the bottom of the ocean at the hydrothermal vents, you know, of the mid-Atlantic ridge, basically volcanic, super hot. The hot springs in Yellowstone National Park, bacteria live there. Why? Their enzymes work at that temperature. That's where they evolved to work. So that the optimal temperature for these two enzymes is quite different between a human and a bacteria. This is a thermophilic bacteria. It can function quite well in those hot areas because their enzymes are adjusted. Now, what's interesting is when we do the DNA study, where we're going to split your DNA up, we restructure it by using enzymes and thermal cycling. So we denature your DNA, make 2 new strands, and anneal it again. So hot, DNA separates, cool, the new strands come together, it goes hot, cool, hot, cool, hot, cool. Well, enzymes have to work in that environment. Human enzymes can't work in that environment, it's too hot. So we actually have kind of borrowed enzymes from bacteria to do that job. And there's an optimal pH. So pepsin is a stomach enzyme. It only works at a very low pH. Any other pH would probably denature it. Yeah, trypsin, that's in your intestines. Well, that's much higher, pH of 8 or so. Very basic, the basic pH. Very important for enzymes. So we'll be doing a cool experiment, not next week, but the week after, on enzymes, determining something about what are known as cofactors. So enzymes do not work alone. The reason that you need minerals and vitamins in your diet is because they are either coenzymes or cofactors. So a cofactor is a mineral. It's a non-protein enzyme helper. A coenzyme is a vitamin. It's an organic cofactor. And these help enzymes so some of them are what are known as competitive inhibitors. They'll compete with the substrate. So here's the substrate, and it's it's going to bind to that enzyme, but a competitive inhibitor comes along and says, nah, that's not going to happen. And that's a way of regulating how much that enzyme is going to work. So that's a competitive inhibitor. Enzymes can't keep working indefinitely. They must be regulated in some way. So that's often by what's known as a competitive inhibitor or a non-competitive inhibitor. A non-competitive inhibitor, here's one right here. It binds elsewhere, but it does change the shape of the enzyme enough that it can't function anymore. So take a look at the active site. It's not going to fit. Yeah. So that's another way of regulating how much product one gets from your enzyme. So it helps control metabolism, regulation of enzyme activity. They must be regulated. Why? Because we're orderly individuals. How are they regulated? Well, one type of regulation is called allosteric, in which a protein's function at one site is affected by the binding of a regulatory molecule at another site. So that would be the non-competitive cofactor. Many enzymes are allosterically regulated. They may be activated or inhibited.
I'm sorry,
Yeah.
is there any chance we can repeat that one more time? I didn't quite.
Yeah.
Sorry. It was
No worries, no worries.
like I heard the words, I just didn't process it fast enough.
That's okay.
No, that one,
Try,
the allosteric regulation.
I'll try to remember. Okay, so allosteric regulation. So when a protein's function is affected by the binding of a regulatory molecule at another site. So you know
Okay.
how, well, for hemoglobin, I think that's a good example, sort of. So you know how there's 4 subunits to hemoglobin, and then oxygen binds to one area, just one heme group, but it kind of tugs on the whole molecule. The whole molecule changes shape. So that's a kind of change of shape. It's not the perfect example. So an allosteric regulation would be this kind of competition. So if this enzyme has made loads of product, it's time to stop, then this non-competitive inhibitor will bind here. This is a site away from the active site. And that's what we mean by allosteric regulation.
Okay, so that is a type of non-competitive inhibition?
Yes.
Okay,
Yes.
perfect.
It's
Thank
not
you.
necessarily that with the inhibitor, yes, but it can also function to make the shape more conducive to binding. It does go both ways.
Perfect. Thank you.
Yeah, if you need
Sorry for
more,
asking too many questions. Yeah.
it's great. You know, any question that you have, I think other people have as well, you know? So terrific. Yeah, thank you. So yeah. So here is a pretty good example. Let's look at it closely. Here's an activator. This little green guy here, an activator. So when it binds here, it's more active. It's the active form. It stabilizes the active form so that it will continue being active. So this is the active form here, stabilized by this particular molecule, whatever it happens to be. Maybe it's a mineral. Yeah. So that's the activator. This is the inhibitor. The inhibitor lands here and it stabilizes this inactive form. This is the inactive form with the, you know, it's just not going to work. Nothing will bind there. Yeah. So there's an oscillation between active and inactive, and it depends on what is required. Is that enzyme required to work or not? So cooperativity is not inhibition, it's allosteric regulation that amplifies enzyme activity. So here's the inactive form here. Here's the molecule that will bind to the site and make it into a stable active form. So like any feedback system, it's often a negative feedback system or inhibition. There's loads of product. The end product may shut down the pathway with feedback inhibition, it's called. So here you've got a substrate. In this case it's threonine. It lands on the enzyme here. It has several intermediate forms and enzymes that catalyze the intermediate reactions. Our end product is isoleucine. So this is an amino acid that we require to make proteins. So we synthesize it. But do we need loads of it? Not really. The isoleucine will bind to the allosteric site away from the active site, but it changes the shape. Look at this. Changes the shape. And now inhibits the enzyme from making more. Then when the isoleucine is used up by the cell, they become available to make more isoleucine. Lots of times those enzymes are grouped into complexes and they're incorporated into membranes in both cellular respiration and in photosynthesis. And they're contained inside organelles. So if you were to look inside mitochondria, there's loads of cisternae or internal membranes, and these are all studded with enzymes. They're embedded right into the membrane. And that's where the sequence of cellular respiration takes place. Which is our next topic. So I'll stop now.